Hey there. Welcome to Science Negotiator. In this video, we break down the different types of tests and highlight some of the new tests that are slowly being available for detecting coronavirus and the disease it causes, COVID-19. Most coronavirus tests that are done involves PCR, which stands for Polymerase Chain reaction. These tests starts with a long cotton swab, which is inserted inside the nose and collects mucus, saliva, bits of cell, and if present, viral RNA. This is then processed and transcribed into DNA, which stands for deoxyribonucleic acid. The obtained DNA is placed in a machine called real-time PCR. It heats and cools the samples in a process that essentially Xerox and make thousands of copies of any genetic material that are present in the sample. These are then introduced to fragments of DNA found in the coronavirus. If any viral genetic material is present, these fragments bind to it. Chemical markers added during this process produce fluorescence when this binding occurs. Two specific genes are observed during this process. A sample is considered positive if both genes are binded, inconclusive in case of only one gene, and negative for none of the two genes. Another type of tests which are being done are known as serological tests. These involve looking for antibodies which the body has produced to fight off the coronavirus. Such tests are more preferable because they can detect antibodies even if a patient has recovered, which is not possible through PCR. These test kits utilize a small amount of blood sample instead of nasal swabs. It uses a special strip which detects two types of antibodies which are produced by the body in response of the coronavirus. The strip contain red blood cells of certain animal species which are infected with a mixture of certain types of viruses. These viruses make the blood cells clog together, which gets unclogged on putting a small amount of blood sample having COVID-19 antibodies. These tests are faster and portable but less accurate than the PCR. However, both the tests might miss if the samples are taken too early when the viral load is low or the body hasn't yet produced the antibodies. Research is being done on a variety of new tests called the rapid PCR. These tests are able to produce the result within a few minutes. They use a set of proteins which amplify the viral genetic material without the temperature changes which is needed in a traditional PCR. This could cut down on the costs and allow establishing point-of-care test centers in a large scale to provide quicker answers to individual patients. While the rapid PCR and serological methods are easier to carry out for small number of samples, the traditional PCR machines allow a large amount of samples to be tested at a time. Based on the density of the infection among the population, each of these methods are useful in their own ways in different scenarios. Comment what do you think down below. Hit the like button and subscribe to the Science Negotiator for more such videos in the future. Click here to watch more of our videos. As always, stay happy and keep learning.